I'm going to bring up our next speaker, Dr. Jarod Roland. He is a neurosurgeon at WashU. If I'm not incorrect. Yes, I'm correct. And I'm really excited to hear his talk. And you should be able to share, so go right ahead.
Perfect. Thank you. Can you hear me okay?
Yes.
Perfect. Let's see, I'm starting to share my screen here. Make sure that's coming up. Okay. Well, very good. Thank you for that. And thank you for inviting me. Thank you, Chad, for that awesome talk. I'm going to walk through a few things that we're working on one of our research studies here at WashU. Some of it relates to some of the things that Dr. Sylvester was talking about kind of towards the end of his talk there. I'll try to tie things in just a little bit. And this isn't a lot of finalized analysis. This isn't things that are published yet. This is kind of interim analysis, but some of the kind of progress that we're making. If you've heard some of our talks before, you've heard us talk about this ongoing study that we have called the Redefining Chiari study. It's something that you've heard us talk about even earlier today, where we feel like the definitions and some of the things that we describe when we talk about Chiari malformations are potentially long overdue for being redefined, hence the kind of overall arching goal and name of the study. You'll sometimes hear me talk about Project 2. I apologize if I slip that in there every now and then because it's kind of common for me to say that, but most other people have no idea what Project 2 versus 1 versus 3 is. But one of the reasons why I mentioned that is of the many projects within this overarching Redefining Chiari study, the particular one I'm talking about today is how we're using some of this resting state functional connectivity. This is some of the resting state fMRI that Dr. Sylvester just briefly talked about, and how we're studying the effects that Chiari malformation has on these networks. So I'll step through this somewhat quickly. This are some disclosures, kind of routine grant funding and things like that. And give a little bit of background. Dr. Sylvester mentioned this earlier, but just a quick overview for anybody who hasn't heard this before about what resting state fMRI is. Sometimes we differentiate this from what is kind of typical fMRI, or what we'll sometimes call task-based fMRI, where somebody might be sitting in an MRI scanner and that scanner is watching we'll say activity of the brain while they're doing some kind of task. What was discovered years ago was that in between these tasks, the brain is active at rest, where previously that activity was thought to be kind of background noise and was regressed out of the analysis. Somebody really smart figured out that there is some structure and there's some meaning to that. The way that that was initially done is a classic task-based fMRI, where somebody's sitting in an MRI scanner and they're opening and closing their hand. And by watching that activity, you could say, here is the area of the brain that's responsible for opening and closing your hand. That was the task that was being done, and that was the kind of correlation that was determined. What somebody did is, in between those periods of moving their hand, when a participant is sitting in the scanner at rest, they analyzed those signals from the different areas of the brain, more specifically that area of the right hand and the left hand. And even though they weren't moving their hands at that time, they saw that those signals were strongly correlated in time. And as you did that in multiple areas of the brain, you saw that various areas of the brain that were participating or responsible for some kind of common activities, some common cognition, some common tasks maybe, were correlated in time. And so that's kind of what we have on the screen here. As you can see, two circles, we'll call these regions of interest. And these blue circles have a tracing over time. And that's what the figure is on the right. This bold change, the bold signal is that correlate of brain activity, and you can see it kind of goes up and down over time. And you see these two areas of the brain have a really strong correlation. When one's going up, the other one's going up. When one's going down, the other one's going down. And this is what we'll call functional connectivity, or one definition of functional connectivity. It's these two areas of the brain that are seemingly disparate in space, but related in function. These two specific areas make up what's become known as the default mode network. This was a really interesting network in the brain that really wasn't even considered before some of this type of imaging. And there's a variety of different ways to define these networks. This kind of table over here on the left is kind of an example of that. This is a small subset of the many different ways you can divide up the networks in the brain. But over here on the right is one of these we'll call parcellations, the way of dividing up these networks that a lot of us here, particularly at WashU, use pretty frequently. You might recognize this figure looking very similar to the one Dr. Sylvester showed earlier, because it's a very similar network. I believe parcellation, I believe it's the one he used. But this is one way of dividing up all of these networks. And this big red region here, this is what we call the default mode network. These represent those blue circles we saw on the previous screen. It turns out when you divide up all these networks, there's different ways of naming these things, and some of these names have changed over time, actually. For instance, this one that's called singular opercular network, there was a recent paper saying that that should probably be called the action mode network to differentiate it from the default mode network. So this is a topic of ongoing discussion. But the big picture, the take-home message is these are the ways that we divide up the brain, calling them into different networks to help understand how these different regions of the brain are functionally related. Here's a little bit of a summary graph. This is way too much to read, but it's the kind of thing where if I just kind of reference you this reference down the bottom left by Ben Seitzman and others, it was kind of a brief overview, kind of high-level picture of what a lot of these different networks represent. One of the things that we wanted to do was analyze how some of these networks might be affected in individuals who have a Chiari malformation. What we did first was we looked at a large study that's already been published where a lot of data has been shared, and that we happened to identify a subset of individuals who had a Chiari malformation in the study. What this was is what's called the ABCD study, the Adolescent Brain Cognitive Development study. It's a really great study. It's ongoing. It started several years ago and enrolled just shy of 12,000, a little over 11,000 kids between 9 and 10 years of age. Got a lot of data on them, tons of questionnaires, and a lot of resting state fMRI data. And the really cool thing about this is it's been funded by the National Institute of Health is they release this data periodically as it's been collected, curated, and processed. They release it for other researchers like us to analyze. One of the really cool things that was done is part of what we call safety reads, while the radiologists who were reading these MRIs as they were being done, making sure there wasn't something unexpected like a brain tumor or something else that might have shown up there that an individual would want to or need to know about. They were wise enough to keep track of incidental findings. And so they noticed that there was about 71 individuals in this data set that had what they called a Chiari malformation. I'm not going to go into exactly how they defined that. There's some nuance there. But it led to a really great opportunity for us to take a look at this data and see if we could find some effects of a Chiari malformation in what's essentially incidentally found or subclinical Chiari malformation in this cohort. And because we have such a large comparison group to compare to, we could do some really interesting analysis from that control group. So we did that. We applied what's known as this Gordon parcellation. It defines 11 resting state networks and divides up those networks into parcels. So for each network, there's more than one parcel. There's about 333 parcels in the cerebral cortex. I like to emphasize that because I find it interesting that we are analyzing how this malformation, Chiari malformation, how this malformation in the cerebellum affects the networks in the cerebrum. Some people think that's interesting. Some people think it's unusual, but we think it's really important that this hindbrain malformation, this tonsillar ectopia that defines a Chiari malformation, probably has significant impacts on our cognition, certainly within the cerebellum and beyond. For this particular analysis, we were focused just on the cerebrum, not looking at the cerebellum, but looking at those cognitive networks. We analyzed 55 of those 71 individuals who were identified with Chiari malformation due to some of the data analysis issues. Not all 71 were available to us. And we selected about 2,000 individuals separate from those with Chiari malformation as a control group to compare to. So when we did that, we looked at all those different networks. And I have a graph like this for all the different networks, but I'm kind of going to skip through a few and kind of point to a few of what we think are potentially relevant. Some of them had pretty significant effect sizes. So this is the singular opercular network. When we looked at the Chiari group compared to the control group, that's what the figure is over here on the left. You can see significantly lower functional connectivity within that network. So how all those parcels within the singular opercular network were functionally connected to other parcels within the singular opercular network. That Chiari group is on the left, and that control group is on the right. And again, you don't have to necessarily look at all these numbers. One of the numbers that's kind of a take-home message is this Cohen's d kind of effect size at the bottom. It generally ranges from 0 to 1, and something on the order of 0.45 is actually a pretty large effect size. But again, the number itself isn't entirely relevant other than to give you a sense of scale or magnitude. So it was a singular opercular network that was pretty significant. The default mode network, again, that turns out to be highly relevant in a lot of neurological disorders. And we think is probably pretty important for a Chiari population just because its role in things like attention or other kind of processing. The dorsal attention network, the frontal parietal network, this is one that Dr. Sylvester was mentioning as well. Again, another one that we think is probably really important in our Chiari population just because of the higher order cognitive symptoms that we hear a lot of our patients tell us about, and the type of cognitive function that's been most attributed to the frontal parietal network seem to have a lot of overlap. So kind of both the DMN, the default mode network, and the FPN, the frontal parietal network, were two of our really a priori hypotheses of what we thought was going to be most affected. And again, this kind of 0.34 is a pretty significant effect size. I'm going to skip through a lot of these just because I went through every single network kind of showing them. I put them all in here just for kind of completeness sake. This is one to show, well, not everything was affected. The sensory motor network can be divided into actually a lot of different ways. One of the biggest divisions is kind of a lateral medial or a hand or mouth version of it. And that network wasn't really affected compared to control groups. The ventral attention network, that was, I believe that was the other one that Dr. Sylvester, I apologize, I had mentioned, again, a pretty significant effect size there, showing some big changes. This is when you looked at those effect sizes, just kind of one big graph. We try to use similar color scales, particularly if you're using this Gordon network, so that the colors in Dr. Sylvester's slides are very similar to these. But you can see how this ventral attention network, this default mode network, and this central cingulate-opercular network were some of the larger ones affected. This kind of RTP in the middle is probably a little bit less important, even though it stands out as the largest. It's not one that we're really going to focus on for a few kind of technical reasons. But this was kind of that initial analysis. What's really interesting about the ABCD study is that this is the analysis from what we call the baseline data, the year one, when all these individuals at 9 to 10 years of age. We're now several years into the ABCD study, and the subsequent timelines have been released. So we're going to repeat this analysis not at baseline, but repeat it at baseline and the subsequent time points to make sure it holds up, to make sure it's not a kind of a fluke thing that happened at the baseline but wasn't repeated over multiple years, to make sure it is truly an effect that's related to the Chiari malformation. And what's probably more impactful is that we have designed our Redefining Chiari study to match the data collection in the ABCD study. So what we're doing is we're collecting similar types of data, the exact same sequences that were done in the ABCD study in our patients who come to St. Louis Children's Hospital or Barge's Hospital, come to WashU with a Chiari malformation, and who were kind enough to participate in our research study. And we're analyzing in a very similar way. We're getting very similar NIH toolbox type questionnaires. We're also getting some more things that weren't included in the ABCD study, or things that have been previously validated for Chiari malformations to dig in a little bit more detail, things specific to the Chiari study, or sorry, to Chiari patients. Because the ABCD study, that wasn't the goal, right? They were kind of analyzing a broad range of things. And the fact that several individuals had a Chiari malformation was essentially an incidental finding that wasn't planned. So we're going to be repeating that in our data. We've have 55 individuals at least in the ABCD study. There's potentially 71, just depending on a few data quality issues. We've enrolled already over 80 from our study alone, and we're aiming for about 150 by the time of the end of that study. And we're going to replicate this analysis and find out what kind of things are truly related to Chiari malformations, which ones are robust to the type of imaging or the setting that it was done, and potentially even robust to age. Because again, this ABCD data set that I just showed was very, very uniform. All these kids were 9 to 10 years of age. We'll repeat that over time as they age throughout the study, and hopefully show that it's robust to their age, or maybe even find things that change over the course of their aging. And then for our patients, they're not incidentally found or subclinical patients. These are the individuals who are coming to us for treatment, and we're obtaining imaging along the way. There'll be a subset of those who are clinical patients who might even have surgery or Chiari decompressions, those posterior fossa decompressions with or without duroplasty. And we're going to repeat that imaging after to see how some of these networks might change with treatment, hopefully for the better. Hopefully, as those individuals have some improvement from treatment, we'll see how these networks may or may not be affected. We think that this is going to provide us a lot of insights into the both mechanism of the disease, but also to help us prognosticate things. As a lot of people on the call probably know, one of the challenges for us is figuring out which of these symptoms are directly attributable to this tonsillurictopia and which ones can benefit from intervention. The current standard kind of anatomic imaging doesn't provide us those answers. So we're hoping some of this advanced imaging, some of these functional connectivity, metrics might provide us better insights to help patients in the future. So I want to thank everybody. That was a really quick kind of whirlwind breeze through what we're doing, kind of what we've done so far and some of the data that's available to us, what we've collected so far, and how we're going to be applying that to the data that continues to be collected. We have a huge team in the Redefining Chiari study, and obviously none of this could be done without those individuals who come to us for treatment and are kind enough to volunteer their time and brains for our fMRI studies.
Thank you, Dr. Roland. That was awesome. I have 1,000 questions, but I am really glad actually that I waited to ask this one for your presentation because I think it's hyper relevant. There was a question about why anxiety might be so prevalent in patients with Chiari. So could you maybe speak a little bit to the network that you found that might be kind of explaining that connection there?
Yeah, that's a great question. Might even be worth bouncing this off of Dr. Sylvester as well, because which network is most relevant to anxiety is probably an open-ended question, or maybe incompletely answered question. Dr. Sylvester might know this a little bit better than I do. And that is one of the challenges with this kind of resting state network approach is it's fairly young. It's had a ton of investigation over the years, so we're learning more and more about it. But it's actively evolving. Like I said, some of these networks are kind of being renamed. Some of them are being divided up. There's a little bit of controversy between what you call a salience network or a singular opercular network, or some people calling it action mode network. Other people call it the language network. And how you parcelate these things can be challenging. And it turns out how you parcelate them when you're doing your analysis probably impacts the results that you find. So it's challenging is a short answer. Which network might be the most relevant to anxiety? I might kick that one over to Dr. Sylvester because he'll probably know it a little bit better than I do.
Dr. Sylvester, do you want to comment? If not, it's fine.
I probably surprised him to
Yeah. Asking him difficult questions.
Yeah.
No worries.
Caitlin, you need to unmute him.
Oh no. Okay, hold on one second. Hmm.
Can you guys hear me okay?
Yes, yes.
Yeah, so I totally agree with Dr. Roland. I think it's an open question, which parts of the brain, which networks are going to be most relevant. The only thing I would add is that, again, anxiety is the most common mental health problem in anybody. So it's already very common. And it does seem like anything where the brain is more stressed then increases that risk. So for example, kids that are born preterm, prematurely, they have increased rates of other sorts of mental health problems, but especially anxiety, right? And so I think that it's probably that anything that has additional stress on the brain is going to be likely to increase rates of anxiety in a non-specific way.
Excellent. So I'm going to ask this one. So you talked about you're doing this with your clinical cases as well, and that kind of opens the question because, incidental Chiari doesn't necessarily have symptoms. So are you going to be able to then compare your clinical cases, particularly your surgical cases, with those incidental ABCD findings and the controls and see if there's any changes in between those groups as well?
Yeah, that's exactly right. One of the benefits of the ABCD, because it's such a large study, so many individuals, over 11,000, and they're doing it longitudinally. So what that means is over time, we'll have samples from 9 years of age all the way up to, I think, 20 years of age, if I remember correctly. And so we'll be able to age match a significant portion of our clinical cohort. There's a few other things that you could potentially match on. Gender and a variety of other things are probably important, but trying to match those things up as close as possible and compare them will be really important for us. One of the things that we won't be able to do quite so well is dig into some of the symptoms that are most relevant to individuals with Chiari malformation in the ABCD study, because that just wasn't what it was designed to do. There are some questionnaires, like I think it's something relatively simple, like do you have headache? But doesn't dive into what kind of headache that is. Is it posterior or holocephalic or band-like, or all those different types of ways headaches can present, and nuanced and probably relevant to individual Chiari, just weren't part of the design of the ABC study, understandably so. But in some of the things that we do for the Redefining Chiari, we do dive into those details. So those things we won't be able to compare, but certainly some of the resting state fMRI metrics we will. Part of the Redefining Chiari study also is, and again, most of the people on the call know this, not everybody who presents clinically with a Chiari malformation needs or will benefit from a surgery. So part of the RC study is we expect certain individuals to get a surgery, and some will be kind of monitored non-surgically. And we're going to get longitudinal imaging in both of those groups. And so that'll kind of serve as a control group as well as we compare the before and after surgery to those who are clinically presenting longitudinally, but maybe didn't get surgery, to hopefully see how intervention might change some of those networks. And so that'll help us hopefully validate some of the things that we see in the ABCD, or maybe rule things out as maybe metrics of non-interest.
Awesome. And then I'm going to squeeze one more question in, and then we're going to get our last speaker. So we're talking about anxiety and stuff like that, but the connection with, agitation, irritability, I don't know if that's something that either you or Dr. Sylvester can speak to about those connections there, and then what you found, how that might be impacting that kind of issue as well.
Yeah, I don't have any findings on this one right now, but it does recall one of the prior talks we had about this this idea of pathologic itch, some of that kind of activation that happens, and that's something that's been one of these things that has come up as we've learned more and more about our Chiari malformation symptoms, and it comes up in meetings like this where we hear from patients sometimes outside of the clinic in some of these groups where individuals can talk to each other and say what they experience, and then somebody else says, "Yeah, I experienced that too." It's like, "Yo, yeah, I experienced that too, and I didn't even know to mention it." And that's one of those that kind of comes up and it relates to that aspect. Sometimes it's a similar concept but manifests in different ways, and that's one of the challenges of defining all these things. Is and I'm so glad that this is what Dr. Seltzer started off his talk about, of how you define anxiety, because otherwise it gets really confusing about what we're talking about. And what one person describes as irritation, somebody might describe as activation, and somebody else might describe as a pathologic itch. But those are really important concepts that are very nuanced, but require a high level of scrutiny to be able to tease apart.
Okay. I don't know if Dr. Sylvester wants to comment, but in the meantime, I do want to get Oh, go ahead.
I can. I mean,
Yeah.
I agree with everything Dr. Roland said. I mean, it's tricky when people come in with specifically about agitation or being upset or suddenly angry or irritated in different situations. It's another situation where it's really important to figure out exactly what's going on, like what's triggering it, what are the situations in which it's arising, because it can happen from an anxiety problem, but it can also happen from ADHD. It can happen from just general dysregulations of mood, where mood is just more labile and regardless of what the situation is. And so that's why it's another reason why I do think it's important, if you're thinking it's anxiety, to really think if it falls into a category that cleanly hits into anxiety about one thing in particular, about a category of things, because it does change sort of even the medicine that we would use if we're trying to help someone with agitation or irritation, right? Because each of those things that I mentioned would have a different treatment. So I don't know if that's helpful, but that would be our approach for how we would think about it.
Oh, that was excellent. Well, thank you both. These have been excellent so far.